Good morning, class. So last time we have started a chapter that is anticonvulsant agent, which is also called as anti-seizure, as well as anti-epileptic drugs. So we have seen the definition, the basic information regarding these convulsions. So these three, it is a disorder in which there is a rapid firing of nerve impulses takes place. So the frequency of the electric discharge increases during these conditions. So these conditions may be the hereditary, congenital, it may be due to the some drug withdrawal effects. So such factors it will affect to produce the seizures. Okay. So we have seen some mechanism how this rapid firing takes place. That is the if the nerve impulses or the nerve membrane is in the polarization, repolarization or hyperpolarization state, there is a resting state, that is no transfer of nerve impulses from one cell to another cell. But when it becomes depolarized state, then there is a transfer of impulses from one cell to the another cell takes place. Okay, that is the action potential when it is in the depolarization condition. So we should know rapid firing means the cell remains more in the depolarization and there is a more action potential takes place. So if we have to treat such conditions, we have to maintain polarization, hyperpolarization or repolarization state so that it may be in resting state. There is no transfer of nerve impulses or there is no fast transfer of nerve impulses must be takes place. That's why what we have to do, as we have seen, inside negative charge, outside positive charge. Okay, so what we have to do, we have to stop the entry of positive charge ions from extracellular to the intracellular. We have to stop this entry, or we have to facilitate the entry of negative charge ions from extracellular to the intracellular. So this is two ways. Either we can inhibit the entry of positive charge ions inside the cell, or we can facilitate the entry of negative charge ions inside the cell. Okay, and opposite, we can, so there is entry of negative charge, exit of negative charge ions from inside the cell to the outside cell, that we have to stop. And the positive charge ions, if they are exiting from the intracellular part into the extracellular part, so we can allow. So these are the four ways by which we can maintain the proper polarization, hyperpolarization or repolarization state, that is the resting state. And in the resting state there is no transfer of nerve impulses, hence we can treat the conditions like seizure, epilepsy and convulsions. Now these are some drugs. Okay, we have already discussed these three parts. So we have to inhibit the sodium ion channel. Okay, so that there is no entry of positive charge from extracellular into the cell. So drugs like phenytoin, carbamazepine, valproic acid, lamotrigine, which comes under these drugs. Next one, negative charge we can facilitate. So we can give the ergonis, that is the GABA mediated chloride channel opener facilitators like barbiturates, benzodiazepine, what we have seen in the sedative hypnotics. Then vigabatrin, valproic acid, gabapentin, tiagabine, all are there in the syllabus which act on the GABA receptor. Then third one, you can block positive ion channel, that is the calcium ion channel. Examples of ethosuximide, trimethadione, and valproic acid. Out of this drug, valproic acid, that is the valproate, is a common drug. So it can act by this mechanism as well as this mechanism. Okay, so this is in brief diagrammatic mechanism of action of all these drugs. Just go through it. Okay, here you can see sodium channel blocker. So you have an active inactivation gate. Generally, there is activation of sodium channel takes place, entry of sodium ions, then again inactivation. So entry is block of sodium ions takes place. Okay, so we can increase the inactivation of sodium channel so that there is no entry of sodium ions inside the cell. So this inactivation gate, the drugs can bind and it will prolong the inactivation of sodium channel. This is GABA mediated facilitator, so they can increase the GABA mediated chloride channel opener. They can increase this chloride channel. So this is present along with GABA. So GABA is one of the neurotransmitter. Okay, aminobutyric acid. So like this, the benzodiazepine binds to the GABA receptor, barbiturate binds to the GABA receptor. So these two parts, that is the benzodiazepam and barbiturates, they are actually responsible to bind with the GABA receptor. They increase the frequency as well as the timing of opening of GABA receptor. So more amount of chlorides can be entered into the cell that we have studied in the sedative and hypnotics. These drugs, vigabatrin and valproic acid, they are responsible to inhibit the GABA T. Okay, that is the transaminase. This enzyme is responsible for the metabolism of GABA into the SSA, that is the succinic semi-aldehyde. So this transfer is inhibited so that GABA remains as it is. Then gabapentin, it will act on GABA. Then tiagabain is also responsible for the GAT one, that is the GABA transporter, that is it inhibits the transport of GABA. Okay, so this act on GABA, that means they will act by different mechanisms to the GABA and they will facilitate the entry of chloride ions. Okay, so GABA is the neurotransmitter. It binds to the GABA receptor. It will facilitate the chloride channels. So barbiturates, benzodiazepam, they binds to the GABA receptor. But other drugs, vigabatrin, valproic acid, gabapentin, tiagabain, they act by other mechanisms. What they do, they are inhibiting transportation, they are inhibiting the metabolism of GABA so that GABA remain as it is. So more amount of GABA can be bind to the GABA receptor and facilitates more amount of chlorides to enter into the cell. Okay, so these barbiturates, benzodiazepam, they binds to the GABA receptor. These vigabatrin, valproic acid, they inhibit the GABA transaminase, that is the metabolite which is responsible for the metabolism. Tiagabain is responsible to inhibit the GABA transporter, that we will see detail later on. Then third one, that is the calcium ion is there. So when channel opens, calcium enters into the cell. But these drugs, they are responsible to inhibit the calcium channel. So there is no entry of calcium inside the cell. Okay, so in this way the mechanism takes place. So that we have seen. Then their classification, phenytoin, phenobarbital, these drugs, they are the classical drugs which are using generally. These are the newer drugs which are newly introduced into the market for the more benefit and more activity. Next part, classification of anticonvulsant drugs, that is hadantine derivatives, oxazolidine derivatives, benzodiazepam, dibenzapine, GABA analogs, and other drugs. So this is the classification. So classification can be done like this one. Classification can be done like this one, or classification it can be done depending upon their mechanism of action. So by these three ways the classification is done. So generally you can write down this classification in the exam as well as the this classification. So this is depending upon their structure, that is depending upon their mechanism of action. Then we have seen a general structure for the anticonvulsant. So you can find out that this is the general structure for anticonvulsant. So X, we can replace X by different functional group or different atom. Then it will lead to the formation of different class of the anticonvulsant agent. So X is NH, that is NH. So that it becomes the hadantine. So it becomes the hadantine derivatives like phenytoin. Okay. So if X is oxygen or X is replaced by oxygen, it becomes oxazolidine diol. So oxa, nitrogen, so zolidine, and two ketones. So it becomes diol. If it is carbon, that is CH2, it becomes succinimide. Okay. And if it is six member ring, that is CONH is added instead of X, so it becomes the barbiturate, that is the six member ring. So in this way, the classification of anticonvergent is done depending upon their structure. As they are having the similar type of structures, only small changes takes place, which leads to the different structure. N means hydantine, oxazolidine, oxygen, succinamide, carbon, and if six membering in which amide group is attached like CONH, it becomes barbiturates. Then what is general ACR? These two substitutions, R and R dash, if they are lower alkyls, then they are active against absence seizures, but inactive against grand mal and partial seizures. If one of these is phenyl ring, that is aryl ring, then activity is directed towards the tonic clonic, that is generalized tonic clonic and partial seizures. लक्षात आलं. सो तिथं जर अल्किल ग्रुप असेल, absence seizure, तिथं जर one of group is aromatic or aryl group, it becomes active against tonic clonic and partial seizures. Okay, so this is the first class of the anticonvulsant agent, that is hadantine. This is the general structure of hadantine. Okay, as we have seen, X is replaced by NH, it becomes hadantine. Okay, so we can substitute three positions in the hadantine. One is R1, R2, and R3. So there are three drugs there for our syllabus, that is the phenytoin, in which this R1 is H, one of R2 and R3 both are the phenyl ring, so it becomes phenytoin. Okay, so what we are seeing, if aryl group, it active against tonic clonic and partial seizure. So phenytoin is active against tonic clonic and the partial seizures. Then second one, may phenytoin. So may means Ame we have added. So Ame means methyl group. So at R1 there is methyl group is present. One of this group is phenyl ring, another one is the ethyl ring, ethyl group. So R2 is C2H5, R3 is C6H5, and at the first position there is a CH3 group is present. Then third one, ethotoin. Etho, as the name indicates, etho means ethyl. So at the first position there is a ethyl ring is present. Okay, so these groups, one group is hydrogen, the R5 actually it is R2, and one is R3, one is the C6H5. In this way, the examples of hadantines are there. So this is the basic of hadantine, which are firstly isolated by the scientist Adolph von Baer. Actually, it is accidentally formed. They were studying the uric acid. During that, they found this drug. They are having a close structure with barbiturates. Barbiturates मध्ये एक एक्स्ट्रा ऑक्सो ग्रुप असतो तो ह्याच्यामध्ये absent आहे. So they become weaker organic acid because there is a absence of oxo group. Now SAR, regarding the hadron, small SAR is there. One of these group, actually it is the fifth position. Okay. 1, 2, 3, 4 and 5. So at fifth position R2 R3 यापैकी एक ग्रुप हा फिनाइल ग्रुप असला पाहिजे फॉर द बेटर ऍक्टिव्हिटी. If they having the alkyl group, they need contribute to sedative effect. जर दोन्ही ग्रुप अल्किल असतील sedative effect मिळू शकतो. Some other examples of hadantine which are not in syllabus, but this pyrohadantine, this hadantine, dithiohadantine, and 1,3 disubstituted means here is also substitution present, here is also substitution present. As such case, they having the activity against chemically induced convergence. So convergence हे आपण बघितलेलं आहे how the convergence are produced. Some are due to the congenital, some are due to the toxins. If by toxins or chemically like MPTP, if it is produced, so if we have substitution in this position and this position at both 1 and 3rd nitrogen, then it produces activity against chemically induced convergence. Okay. So this is small SAR regarding the hadantine. So the first drug phenytoin, it is metabolized by p hydroxylation as phenytoin. They having the two phenyl group, both having the no substitution at ortho meta para position. So substitution of hydroxy group can be takes place at the para position, either this one or this one. So they can be metabolized by p hydroxylation, and when they undergo p hydroxylation, this hydroxy group can be undergo this phase 2 metabolite, that is conjugative metabolite. In case of methatoin, on nitrogen they having the CH3 group. So these methatoin they can undergo N-demethylation, and when they undergo N-demethylation, they produces the active drug. So methatoin is the hadantine analog, which is prodrug. Which is the active drug? When methatoin, it will undergo the methatoin. You can structure R1 having the, here there is a CH3 group is present. So when this CH3 group is present, so it undergo N-demethylation. So हा CH3 group निघून जाणार, इथं hydrogen येणार. This is the active drug or active form of drug methatoin. Okay. Similarly, ethotoin, it is also undergo dealkylation into active drug. Then phosphenetine is the phosphate ether. This is the phosphate, it is phosphate ether of phenytoin, which undergo metabolism to form active drug phenytoin. Okay, so the phosphenetine is the metabolite, or it is the prodrug which is undergo metabolism dephosphor into the active drug, that is phenytoin. Okay, so this is the, just go through it, this is the metabolisms of phenytoin. So in this way other drugs also undergo the metabolism. So this is phenytoin. Okay, so two phenyl groups, they are having no substitution, so they undergo hydroxylation. So at para position the hydroxy group is attached, and then it undergo the glucuronide conjugation. Similarly, meta hydroxylation is also takes place and it undergo conjugation. Okay. Or both the meta and para undergo hydroxylation at same time, then they undergo conjugation. Okay. Here also you can observe both the positions meta and para. They having both hydroxy groups. So hydroxylation takes place. Then one of the hydroxy group in undergo methylation to form OCH3. Then their conjugation takes place. So this is the one of the example for phenytoin. So other drugs like mephentoin, ethotine, they also undergo the same type of mechanism to produce the phase 2 metabolism, that is the inactive drug. This is the next drug. So hadantine, we have seen their SAR examples, that is the phenytoin, mephentoin and ethotine. Then their metabolism is important part. Then second class of drug, that is the trimethadone, succinamide. Okay, instead of N there is the presence of carbon, so it becomes succinamide. Derivatives, so example is trimethadone. Trimethyl means trimethyl group. So two methyl groups are present here, one methyl group is present here. So drug is called as trimethadone. Actually, it is a prodrug. It undergo एन डिमिथायलेशन. इथला जो CH3 ग्रुप आहे तो निघून जातो. So it is the एन डिमिथायलेशन and it parts NH, that is the active drug. This active drug is called as dimethadione. So tri means 3 methyl, di means 2 methyl. So it forms dimethadione as one of the methyl group is removed. So it is the calcium channel blocker. Okay, so that is the second class of drug. Hadantine. Next one, this trimethadone. Then already we have seen benzodiazepam, their mechanism of action in detail in sedative and hypnotics. So they are also useful in the treatment of convulsions to decrease the seizures. Generally, diazepam and lorazepam, two drugs are used. Along with that, clonazepam is also used. Lorazepam is not there in the syllabus as an anticonvulsant. You have two drugs, diazepam, clonazepam, as the anticonvulsant drugs under the class of benzodiazepam. बाकीच्या ठिकाणी बेझोडायझेपाम मध्ये याची एक्झाम्पल्स आहेत. बट इन केस ऑफ अँटीकन्व्हल्जंट, डायझेपाम अँड clonazepam, these are the two examples there in the form of benzodiazepam as an anticonvulsant drug. So detail regarding its metabolism already we have seen in sedative and hypnotics. Okay, just go through that one. The next drug, carbamazepine. Okay, so dibenzazepine. Dibenz means these are the two benzene ring, that is the dibenz. This is 7-member pine and having the aza group, that is the nitrogen. So carbamazepine comes under the dibenzapine derivatives. Okay, so that is the nomenclature for carbamazepine. 5-hydroxy dibenzo in bracket BF azepine 5-carboxamide. Okay, at the fifth position, 1, 2, 3, 4, and this is fifth position. Fifth position la CONH group hai. It is called as carboxamide. So 5-carboxamide azepine, as it is the fusion ring, it is nomenclated like this one. So you have studied in the heterocyclic chemistry how to nomenclate the structure. So like this you have to nomenclate the structure. It is useful in the treatment of epilepsy and bipolar disorder. Its structural features similar, MOA is similar with the drug phenytoin, or you can say hadantine derivative. So the main important part regarding carbamazepine is their structure, their name, their mechanism, and important is their metabolism. So there is a compulsory question is asked for 2 marks. Give the metabolic products of carbamazepine. Okay. So if you go through the book, what I told Brahmankar for the biopharmaceutics in phase 1, phase 2 metabolism, they have given examples regarding carbamazepine and its metabolism. So this position, there is double bond is present. So this double bond is broken and epoxide formation takes place. Double bond broke होतो आणि तिथं ऑक्सिजन येऊन अटॅच होतो. So such structure is called as epoxide. So the drug is called as carbamazepine epoxide. Then this epoxide ring breaks and it will give the 2 hydroxy group, that is the carbamazepine diol, as it consists of 2 hydroxy groups. So this is the major metabolism of carbamazepine. Next one, second, it undergo reduction. So this double bond is reduced and hydrogen is attached to these two parts. So it is called as 10,11 dihydrocarbamazepine. Okay, you see numbering. So this is 1, 2, 3, 4. This nitrogen is 5. This carbon 6. This carbon 7, 8, 9, 10, and this is 11. So हे डबल बॉन्ड आहे हा 10 आणि 11 मध्ये. सो त्यातला डबल बॉन्ड रिड्यूस केला. सो इट इज कॉल्ड एज 10,11 dihydrocarbamazepine. So that is the second pathway of metabolism for carbamazepine. Next one, hydroxylation. At this position is free, this group is free, there is no substitution. So at this position, the hydroxylation takes place. So it is called as 3-hydroxycarbamazepine. And at 10 position also hydroxylation may takes place. So it is called as 10-hydroxycarbamazepine. So this question is compulsory asked for 2 marks regarding give the metabolism of carbamazepine. So this is epoxide formation, then breaking of that epoxide, that is the this is the major metabolism, then reduction reaction, then hydroxylation at third position, hydroxylation at 10 position. Okay, so these are the important metabolism of anticonvulsant drug, that is the carbamazepine. Okay, so this is about today's lecture. Remaining drugs we will see in the next lecture.